Okay, now we're at 6.3, which is basically when all the magic happens. So, first of all, when you get, the process of getting the pollen grain to the stigma, that is pollination. The pollen grain going down into the embryo sac and fertilizing is called fertilization. So just note that there are two different processes. One is the transport of the pollen grains from the anther to the stigma. Second is the process of the pollen grain getting into the embryo sac through the micropyle. So yeah, one is called fertilization, the other one is called germination. Pollination. If you, I said it terbalik, but you get what I mean, right? You know which one is fertilization, you know which one is pollination. Okay, so how does it happen? First of all, as I said earlier, the wall of the pollen sac of the anther, it will burst. When it bursts, not burst, it just breaks. I'm not sure on how it breaks because it says here that the wall of the anther from mature pollen will dry, shrink, and split. But basically, the anther breaks and it causes the pollen grains to burst out. So when it gets released, somehow it will get to the stigma, either through its own pollinating agent. So it can either be insects, mammals, birds, water, or wind. The main one here is insects because we all know about the bees. But yeah, that's the main point. That's the main part. The bees. That is the most popular example. But yeah, it somehow gets onto the stigma, and what happens is it will germinate. The pollen grains will germinate. So what happens when it germinates? It will form a pollen tube. So it goes on here, and then suddenly it starts going through the style. So how does it go through the style? What happens is the end of the pollen tube, which is the tube nucleus punya area. So the tube nucleus is here. It will, for some reason, I'm not so sure which one secretes the enzyme, but an enzyme will be secreted. I think it's through the tube nucleus. It will secrete an enzyme, and what happens is the enzyme breaks down the style tissues. So that's why the tube nucleus is digging a hole, because its enzymes are breaking down the style tissues. And then what happens to the generative nucleus here? You see, it's generative nucleus, but how does it become male gametes? So in this part, when it starts going down the style, when it starts generating the pollen tube, when it starts moving along the pollen tube, the generative nucleus will divide by mitosis. So it stays the same number, which is haploid. So it will produce 2 haploid male gametes. And then again, this tube nucleus will continue digging down, and then it doesn't dig down like this. It doesn't go immediately in. It will go through the micropyle, because that's what the synergid cells told them to. So it will go down, it will bend, and then it will go into the micropyle. So when it goes into the when it reaches the micropyle and penetrates into the embryo sac, it will the tube nucleus will be like, okay, my job is done, I will die off. So the tube nucleus degenerates, because it doesn't have to dig a hole anymore. It already reached the location, if you get what I mean. So yeah, the tube nucleus is already done digging. It dies or just degenerates, and then both male gametes enter the embryo sac. So after the tube nucleus degenerates, the male gametes just enter inside casually. So now what happens after it enters inside casually? We call it double fertilization, because there are 2 fertilizations that occur. The first one is the formation of the diploid zygote. So what happens is one male gamete will fertilize with the egg cell. Remember that the egg cell becomes a zygote. So this embryo sac, this egg cell fertilizes with one male gamete. Remember that there are 2 male gametes. So one, it becomes a zygote. How about the other one? Where does it go? Well, remember that I mentioned that it's very important that you know that the central cell has 2 nuclei. These 2 nuclei, for some damn reason, fuses with the other male gamete. So this male gamete goes in here. This male gamete goes into these 2. And remember, each of these are haploid. N. So this is already 2N. And then the male gamete is also N. So it will form a triploid endosperm nucleus. This is very important. the most important part in the chapter is that you know it produces a diploid zygote and a triploid endosperm nucleus, or just a triploid endosperm. So yeah, I think that is the main point you have to know. One becomes diploid, one becomes triploid. Because there are two fertilizations occurring, we call it double fertilization, and it's quite unique. Okay, how about the importance? This is the all basic stuff lah. So why is it so important that we have double fertilization? Number one, the zygote ensures that genetic information is passed on from one generation to the next. That's quite obvious. And then since we form a diploid zygote, when it divides by meiosis, it will become haploid again. So the haploid condition in gametes will be restored, because it will still continue its diploid become haploid thing, because basically, if you get me, Gan, if we were to have just diploids fusing with each other, when you have a diploid and a diploid fusing, it'll become tetraploid, 4n. So this haploid condition, this meiosis is very important to ensure that the number stays the same. The number of chromosomes, the diploid and haploid condition stay the same. That's basically it. So yeah, diploid becomes haploid, two haploids produce become a diploid, and the process repeats itself over and over again. That is what B should be talking about. How about number 2? The fusion of another male gamete with two pollen nuclei produces endosperm tissue. So first of all, the endosperm's main thing is to supply nutrients to the seed. But not all seeds need endosperm. It says here, only a part of the endosperm is utilized for the development of an embryo. Some of them are stored in the cotyledon to be utilized during the germination of the seed. So the endosperm tissue, it will some seeds won't even have endosperms, because the endosperm's nutrients go into the cotyledon. But the main thing here is it still supplies nutrients. So number 1, it supplies nutrients, and then this here enables the embryo to survive in the seed for a long time if conditions are not favorable for germination to occur. I think that's how seed dormancy works. But again, I'm not so sure because I'm not that good at plants. So yeah, even though some don't use the endosperm for supplication of nutrients, they use the cotyledon instead. Eudicots, which are dicotyledons, do use the endosperm. So monocots, they don't use it. Eudicots, they use the endosperm, but the endosperm still supplies nutrients. If I'm correct. So that's why it is very vital in the survival of the species, because it helps with the development of the embryo. But yeah, again, I'm repeating it one more time. Even though the endosperm might degenerate, might not be useful anymore, the main use of it is to supply nutrients. Before the cotyledon gets its nutrients, it gets it from the endosperm. And if it doesn't have a cotyledon, then the endosperm itself will supply nutrients. So the main function of the endosperm is to supply nutrients, and that is very important for the seed survival, because if the seed can't grow, it has to keep taking in nutrients, so the endosperm is where all of those nutrients are stored. And also, before the seed can even develop in the first place, it has to get nutrients from the endosperm. So yeah,